So Biology Club, we host events every second and fourth Mondays of the month. Our next events are going to be on the 12th of April on how biology forms Spartans, and then on the 26th, we'll have how the open science community can help cultured meat. You can join this group by just joining our Biology Club Google group. You have the links here. You can also go on Meetup to check all the events hosted by Makespace and Biomakespace. And of course, if you want to join the community biology chat, you can do so on Mattermost. If you want to get in touch with Biomakespace and chat with all the members. So our host today is Al Edwards. We are having a session about how to bring biological and medical innovations to market. Dr. Al Edwards, he's an associate professor at the University of Reading and co-founder of a health tech startup that aims to commercialize immunoassay diagnostics, more specifically on the development of affordable microfluidics. I'm sure he can tell you more about this, at least better than myself. I had the pleasure to work with him a few years ago, and he was invited here today because I think he has a lot to tell us on how to explore new ideas and just about the current challenges in biotech or medtech. And yeah, overall, I think he might have a lot to say on this topic. And on this note, we can start the presentations. And so I'll pass the word on to you, Al.
Hello, yes, so that was a lovely introduction. And it was a great pleasure a few years ago to have Barbara working in my lab on her master's. She did a fantastic job. And before that, at some point in the distant past, I was brought up in Cambridge, so I know Cambridge very well. I did an undergraduate degree there, and my father, who's on the call, had to disappear for a short while whilst I was taking my final exams. Otherwise, he'd have had to mark my exam papers. So it's all very closely related in the family. Anyway, I also have a grandfather who was at Babrahams even further into the past. So I've got lots of strong connections with Cambridge. I'm just trying to work out where the button is. There it is, so I can share my screen. I wanted to say I'll try and keep the presentation short, but that's always dangerous, so I won't say that. I want to tell you about some of the technology that we've been trying to develop and trying to commercialize. The bit that's really interesting, I'm sure to you all, I feel almost like false advertising with this title. So I've added one character to the title by adding the question mark after the ideas to reality, because I think it is definitely a discussion point. I think if I click on present now, is that now presenting properly? Yeah, great stuff. And can you still see what I'm waving around as well as the slides?
Yeah.
Great stuff. So I think, you know, what I'll be telling you about in my experiences to share, I'm sure many of you have heard before and you're very aware of, because I think it's fair to say, I think we'd all agree that turning ideas into reality is quite difficult. I wanted to raise a few questions, you know, about why it's so difficult. I wanted to, when we get towards the end, to raise the question of whether patenting and IP protection is a good thing or a bad thing, or whether open hardware is a better alternative. And I also have just left on the title heading with this comment about concept or product, which is that I've learned over the years, and to a certain extent, that's the role of academics, I believe, is that it's sometimes much easier to develop a concept than to actually develop a product all the way through. So by way of introduction, I have this slide just in case I'm traveling, which isn't a big problem at the moment, but just in case any of you don't know, Reading is not very far from Oxford or London. I like to think of Reading as part of the Golden Triangle. I was born in Oxford and have a PhD from London, and I'm based in Reading and have a degree from Cambridge. So I consider myself to be thoroughly Golden Triangle background, but perhaps more relevant and more interesting is I actually originally did my first degree in science and biomedical science, and then I did a PhD in immunology. But after roughly 10 years as a sort of hardcore immunologist, I moved more into technology development, and I probably consider myself to be an engineer now. I'm surrounded by tools and things like engineers should be. But that's an interesting transition. It's something I think we all agree we need more of. So it's a really exciting area, and it's an exciting time for it. And I realized I had to redo all of my slides now, because I used to have to explain what lateral flow immunoassays are. And I think in the last year, we've learned quite a lot about what lateral flow immunoassays are. And actually, people are much more aware of the limitations of lateral flow immunoassays. But to start off with, we've got this generic problem of bioassays, whether it's clinical diagnostics or anything else. If it's analytical and it's biological, we can think about lots of different ways of doing it. But if we want to start the simplest way, we used to use, my mother would have used a glass test tube to do immunoassays in infectious disease surveillance. The general concept is you have a sample, you combine it with some reagents and somehow you mix it together. And generally speaking, you get a visual change, some kind of color change. Of course, there are many, many different ways of measuring things biologically, but that's a simple way of thinking about it. And I think it's fair to say, and I think from the introductions, quite a lot of you are very aware of this, that we can expect a sort of a step change in the way we do bioassays because of technology that's been developed recently. One of those is actually in the tube itself. So we've got test tubes here. As I say, my grandmother's generation would have done immunoassays, and they did do immunoassays for infectious disease, things like Ebola and smallpox and all of these different things. They would do an immunoassay in a glass test tube. But about the time I was born, a microtiter plate was invented and became the dominant technology. So for whatever, the past 40 plus years, we've generally done assays in microtiter plates. But I think we're ready to change to the next size down. So we've gone from this size through microtiter plates, which have a volume typically of about 0.1 to 0.2 milliliters, down to microfluidics, which is a massive research field. I think some of the people on this call are already working in this area. Our particular variety of microfluidics looks a bit like this. It's a plastic ribbon that's mass produced, and we've produced this by the kilometer, actually. I think Barbara herself has probably used several tens, if not hundreds of meters of this when she did her master's. And inside it, we have these tiny capillaries, which you can't quite see because they don't focus, but you can see on the screen. They're 0.2 millimeter diameter. You can make them a bit bigger or smaller. And each plastic strip has 10 capillaries. So that's kind of cool. You can imagine that you can use this to do all kinds of different bioassays. And I'll go through some ideas about how we're using this, but I don't want to go into too much detail. So it's going to be a bit of a whistle stop. Because I think the discussion at the end is much more interesting, and I'm hoping it will be a dialogue rather than me telling you, I can tell you all the pains that I've had, but I wouldn't consider myself an expert. So let's think about what kind of bioassays we want to do. I'm jumping around a little bit here. There's a wide range of different things. That's one of the problems if you invent if you invent one of a big family. Of course, microfluidics is massive. I didn't invent microfluidics, but if you invent a type of microfluidic device, you have this almost endless range of different things you might want to do. And over the past few years, I've focused a lot on blood biomarkers and also on bacterial detection and identification. Some of the things that we're really interested in are things like urinary tract infection. I don't know whether we can do a pointer. Oh, yes, we could do a pointer. That's great. So we can think about measuring bacteria in urine. We do a lot of work on blood. More recently, I've got very excited by mastitis in cows. So we have pictures of cows and milk. We've also got one of my favorite old medical slides here, because not that long ago, if you had a problem with gonorrhea, you could just pop into your doctor and buy some penicillin because it cured gonorrhea. And of course, as we all know, antimicrobial resistance is a steady march, and penicillin no longer cures gonorrhea. So we don't have the tools that we need to use antibiotics in the most intelligent way, and this is driving increasing resistance. Some of the other examples in the immunoassay area are things like infectious diseases, which again is something I used to have to introduce and convince people, but I think everybody knows that diagnostics for infectious disease is quite important. So there are lots of grand challenges. All of them revolve around some of the same themes. For example, if we take antibiotics, one of the biggest problems is we want to know when we have a patient with a disease, it's quite easy to diagnose a urinary tract infection from clinical signs, or even by looking for bacteria in the urine. But at the moment, it takes 2 days to detect antibiotic susceptibility, very simply because we streak out the urine on a plate and we wait until we've got colonies. That takes a whole day. And then we pick the individual colonies, we carefully resuspend them and plate them out, and then we measure whether or not they're resistant to antibiotics. But if you wait 2 days, then the patient needs treatment during those 2 days. So what we tend to do instead at the moment is not bother measuring antibiotic resistance at all. We just say, oh, well, you can have some antibiotics. And the problem is that we're not matching antibiotics to the patient. So if there's resistance, that's a problem. And we need to be able to make antibiotic resistance testing much quicker and also more portable. Jumping over to my other area of analytical biology, we have immunoassays. Immunoassays are the most effective way at the moment of measuring blood biomarkers, so low concentration, single targets in a very complex fluid such as blood. These can be very important for things like acute and chronic cardiovascular disease and other conditions. The idea at the moment is that the more biomarkers we can measure in a patient, the more precisely we can diagnose people. But at the same time, we also have massive problems across the world with infectious diseases. One of the really big ones that people haven't necessarily heard of is dengue fever, because the mosquitoes that spread it don't live in this country because it's too cold. But for most of the rest of the world, more than half the world's population lives in fear of dengue fever. And we don't have very good diagnostic tools for monitoring and diagnosing dengue fever for various reasons. So if we think about the way that we do immunoassays, we kind of have different extremes. We've got our currently much loved mass-produced lateral flow test, which, as I say, about a year ago, I would have had to explain what that is. This is a lateral flow test. It's a small piece of nitrocellulose with some antibodies on it and some colloidal gold in a plastic cartridge. The great thing about lateral flow is it's super cheap, super mass manufacturable. It's almost easy to use. It's not really as easy to use as people think, but pretty much everyone in this country is going to use it now. So it's obviously easy enough. This is your pregnancy test or your COVID rapid test that's being used in community screening. And you've also got microplate assays. So these are lab skilled assays. They are much higher analytical performance, and then you've got big machines which can do automatic immunoassays. We know there are some limitations of lateral flow, which I won't go into, but I'd love to chat about it if anyone's interested. One of the biggest things is they're about 200 times less sensitive to targets than we would like, maybe even more than that. So the general picture is that what we want is be able to do more tests than outside a lab, but we want to retain the lab sensitivity. So we can kind of plot it like this. We've got fancy instruments, but they're not portable, but they can measure literally a few hundred molecules in blood. Or you can have these very low sensitivity, analytical sensitivity lateral flow tests, which are cheap and cheerful, and you can mass produce them. Obviously, we're up here. This is where we're targeting with our micro capillaries. And I waved around some of the material, which I've already lost. Here it is. The idea is we turn this long roll into tiny strips, which would be a bit like this. And we can kind of scale up the production of that by mass coating this and then chopping it up into pieces. So that's fine. We have a company that we founded quite a long time ago now to commercially develop this. We have some IP, we've got some patents. We've spent quite a bit of money here and there. Some of it was our money, most of it was grant money to develop point of care readers and automatic systems. We've done a huge amount of work. It's way more sensitive than lateral flow because of the way that the assays work. It's much faster than a microplate. So it's in that sweet spot of high performance, portable miniature. We've tested it now with human clinical samples in Thailand for dengue fever. We've done a 250 sample trial, which is fantastic because it proved that basically this microcapillary material behaves exactly the same as the microplate ELISA, except that it's small and portable. And that holds true even if we image the assay results with a smartphone, so we don't need a lab instrument. So that's all very cool. And we also did lots and lots of analytical microbiology. Sorry for jumping around, but we can detect and identify bacteria in these capillaries. We don't do that using immunoassays. We actually do that using an even simpler method, which is we put the bacteria in the little capillaries and we measure the color change. So we can do things like measure the resistance to antibiotic simply by seeing whether, if we have bacterial growth without antibiotic, if we add the antibiotic, does it kill the bacteria? So that's a really important part of analytical microbiology. So what has this got to do with the bigger world? Well, there's a bigger world of microfluidics, which I won't discuss, but I wanted to touch particularly for this makerspace community and people who have worked in the technology development and engineering and hardware and software area. I wanted to make a point which is probably stating the obvious, but I still think we need to keep remembering this is true. Consumer technology has completely revolutionized the way that we can measure things, including in life sciences. So we have all of this stuff that, you know, I'm sort of starting to feel like I'm getting a bit old and talking about how, oh, when I was a boy, we didn't have this stuff, but it's really not very long ago that we didn't have these phones. And the phones, you know, this phone's ridiculous. It costs, I don't know, one or £200, and it's got more cameras than I can even count. It's absolutely ridiculous. It's probably got a camera I've never even used. It's also got an insanely powerful computer. So all of the components behind this, because everybody's buying these by the million, and there's huge amount of money going into the mass production of these imaging chips, there's so much hardware, electronic hardware, digital imaging hardware, and computing hardware, that it really completely changes the way that you approach analytical biology, I think. And I think we're only beginning to start to see this. And there's also, so there's opportunities in instrumentation, particularly because of these imaging sensors, and that comes from the hardware. But there's also a revolution in computing. Again, I don't need to, this is kind of stating the obvious, but it really is important to understand this is going to completely change the way we do some parts of biology. Because of course the computing hardware allows you to analyze what happens when you take an image in a way that wasn't imaginable even a short time ago. So I just want to give you a couple of examples of what we've done, combining the super cheap, super high performance hardware that's coming out of the consumer electronics situation, and also being able to access this consumer technology, the actual chips and the computers and things. But when you combine that with microfluidics, you can do lots of cool things. So for example, I already mentioned that we can do a standard functional antibiotic resistance test. Again, it's super simple. We've got 10 capillaries. We put our bacteria in the capillaries. In the different capillaries, we have 4 different antibiotics in duplicate capillaries, and we also have no antibiotic. If we have a susceptible strain, the blue color means that the bacteria can't grow in the presence of antibiotic. This only takes a few hours to detect bacterial growth. The white capillaries are white because the bacteria have grown and they've eaten up the blue dye. When you have resistant organisms, they grow in the presence of antibiotics. So that's kind of cool to show the power of the microfluidics. But when you combine it with a smartphone, you can put it all in a box, the whole thing. And this box here is what we call MilkGuard, is a product that we developed in collaboration with vets in India who want to be able to do analytical microbiology, but they don't have a lab, and the lab is a long way away and is really hot and the milk goes off. So if you want to find out whether a cow in India in a small rural farm has mastitis, if they have mastitis, what kind of bacterial resistance to antibiotics they have, they just can't do it. So they're stuck. And the levels of resistance are really high because antibiotics are used on a massive scale by huge numbers of farmers all over the place. It becomes very, very difficult to treat these bacterial infections. And of course, they spread from animals to humans as well. So everything fits into the box. There's an incubator in there, which is super simple. The smartphone can image the results of the antimicrobial resistance test. We've got some bits of cardboard, which we actually think is just as important because we need to show the person how to actually use it. And that box is a product concept. It's been shown and tried in India, but we haven't actually managed to get the clinical evaluation with the actual samples yet. But that comes not because of the microfluidics and not because of the smartphone technology, but because of combining the two together. So it's really, really cool what you can do. what else can we do? I have a PhD student at the moment who's just demonstrated that you don't even need to use dyes. These images here are taken with a smartphone camera of the individual capillaries, and the capillaries are filled with bacteria, and then we shine a single LED light on the bacteria, but then we put it on a black background, and the light scatters from the bacteria. So you can see the bacterial cell suspension, and you can see a concentration effect. And actually, if you look over to the left, if you leave them for a little while, you can even see the bacteria sedimenting, which I think is really cool. Over a 30 to 60-minute period, in that 200 micron capillary, they just settle down, and you can actually see them just with a smartphone, which I think is pretty cool. We're not quite sure what to do with that, but it means you don't even have to use a dye or anything like that. We can also detect the Bacteriophage Lysis, which is what we're showing over here, which is where you don't see any bacteria because they've been completely disintegrated by a bacteriophage in only a few hours. And that's a practical that a biology lab would do with a real test tube or in a cuvette. You get the bacteria, you mix them with the phage, and they disappear, and it's kind of magic. But to be able to replicate that in a tiny, tiny 0.2 millimeter diameter capillary image with a smartphone, I think is pretty cool. So just another example of a consumer-driven revolution. I don't need to tell this audience that you can buy a 3D printer for less than £200 that works perfectly well. And that's based on things like RepRap movement and open hardware. So we used exactly the same hardware. Some of you will recognize this hardware. Only instead of 3D printing, we've got a camera on there, which is a Raspberry Pi camera. And using this very simple robotics, we can now automate our microfluidic system and do very, very high throughput microfluidics because we have a really big bed and we can take pictures of our microfluidic devices every few minutes for as long as we want to. And using this system, we can do sort of mass screening experiments where we have thousands of individual capillaries, and each capillary is only measuring 1 microliter. But if you have thousands of those and you do it in a 96-well format, you can do all sorts of wonderful bacterial, analytical microbiology. So that's combining open source, super accessible hardware. Pretty much everything that went into that, apart from the Raspberry Pi, is fully open source. And even that's almost fully open source. And so we literally take all of these modules that other people have developed, we put them together, we build it up. I think the whole hardware bill of materials is under £500. And then we have a robotic microfluidic analytical microbiology platform, which I think is a pretty cool thing. So just to step back, we've got this combination of different needs. We've got things like analytical microbiology, we've got immunoassays, we can use things like microfluidics, we can use digital cameras, we can use tiny computers. But of course, what you all came here to know is how do you turn ideas into reality? And we have got products which are on the market technically. You can buy them. We're very keen for you to buy them. We're not terribly good at sales and marketing at the moment. We're very much better at developing technology and developing concepts. And another really good question that all of you would say is, why is the UK government buying, I don't even want to think about how much, it's like £10 billion worth of these lateral flow tests, when I've told you that they aren't very good. And they are limited in what they can do. But there's a really simple reason why we're using mass screening using lateral flow, which is that there are factories which build and make lateral flow tests. And these factories can produce millions of tests a month or even millions of tests every week. And the reality is the technology that we've been developing is not mass produced in a factory. And we can ask lots of questions about why that is. So this is where I kind of hand over to you. I've got some ideas and things that questions I wanted to pose or suggestions. I've been through this whole process of, looking at the market, developing concept. We've got to the stage of clinical evaluation, but it's really difficult. There are all these questions right from the start about whether you want to protect your IP or whether you want to go down an open route. Both have their advantages and disadvantages. I've got another slide here where we can break this down. I mean, this is, all over the place. You can learn about this, but the reality is that it gets much more expensive. Every step you go towards products, doing the proof of concept. I mean, the reason I showed you the bacterial light scattering experiment is because a PhD student in her first or second year could get some material, do an experiment, say, look, I can do this. With one LED and a smartphone, I can measure bacteria. But going on from there, the next stage, you have to develop a product. You have to engineer it. You have to optimize it. You have to make it work every time. Then when you go on to the next stage and you want to manufacture it, you need even more money and more investment, more commitment. And even then you can spend a huge amount of money and time, and then you still have to validate it, and you have to validate it in the real environment. And if it's a clinical diagnostic, you really nowadays have to do a clinical trial to prove that the product you've developed actually has clinical benefits, which is not easy. This is more like a pharmaceutical development pathway. And when you get to the end of this triangle here, which looks kind of like it's getting big anyway, then you've got to sell your product. And that takes years and time and a lot of energy and money. And then layered on top of that, you've got things like the costs of running your business. If you're developing a business, you've got patenting costs. So I definitely wanted to thank my funders, of course, because we all like to show off how much grants we've got. I want to thank all the people who've done the work, not to mention Barbara, who's done some very cool stuff I couldn't even talk about, which we're very excited about, measuring blood function and just looking at blood clotting. Whole list of people who've done the work. But at this point, I will hand back to Barbara, who maybe wants to chair a discussion. And I would be interested to know what questions people have about how do you develop these things? Why is it so hard? And if there's anything we can do to make it easier.
So if anyone wants to start the discussion or has any questions, also if you don't want to chat, you just can write your question and I can move on from there. So anyone?
Hi. Mike, am I audible? Can you hear me? Yeah. So you mentioned about liquid handler robot you made with open source components, but why you need to make it again ground up when there is OpenTrans? Can you, is it any inspiration from OpenTrans, or it was done before OpenTrans?
So it doesn't do liquid handling. It's just an imaging system. It's really, really simple. It's just a camera on a XYZ stage, but that was what we needed to do. And OpenTrans didn't at that time do it. There is actually a product you can buy possibly from a different company looking at that particular setup. Essentially, it's just a big camera on a frame. But it really took us no time at all to put that together, apart from actually assembling the system. And the point is that was exactly what we wanted. Even the camera configuration was what we wanted. So we thought the interesting thing was that it's completely customizable. But I agree, yeah, OpenTrans is brilliant. So for liquid handling, we haven't developed anything for liquid handling because, as you say, there's OpenTrans and there's other systems out there. I don't think any of them are perfect. With our system, a lot of the liquid handling is done by the capillary forces with the capillary strips. Essentially, in order to fill the capillaries, the microcapillaries, you just put them into a liquid and they fill up anyway. So we didn't need liquid handling for that particular system.
Okay, thank you. I have more questions, but I'll ask them afterwards.
I was quite interested in the topic you were introducing about patents versus open source. Could you?
Brilliant. Let's go to that slide. Yes, please. Are you going to comment, or you just want me to talk?
Oh no, I just thought I'd give you the question. It's on.
So okay, so the really simple way I see it is that, and I actually heard this discussed by somebody who's thought about this a lot more in this, not necessarily in the biology stroke, life sciences area, and he wrote a very coherent and incredible post saying that the patent system is just like Winston Churchill describes democracy, i.e. it's the worst possible system for managing intellectual property, except for all the other ones. And I think his point was that patents cause immense pain and trouble and lots of difficulties. But there is the flip side of it, is that if you don't have a patent, it's very difficult to persuade people to invest the money that you need. If we jump ahead to the next slide, which I've just said, you're going to need a lot of money before you're going to make enough money back to cover all of your development costs. So I'm an inventor of patents which have been granted in Europe and US. I've gone through all of the different stages. I've raised money to pay for the patenting process. It's kind of cool. I know what those patents actually mean, and almost nobody else does, because nobody really ever reads them. And it strikes me as interesting. So the other thing about patents is that licensing them is very difficult and complicated. But I've talked to quite a lot of people over quite a few years now about this, and I'm not convinced that anyone finds patents easy or very successful. In a few circumstances, people say, well, I have this patent and we made all this money, it's brilliant. But almost all the other people I talk to, it's just been pain. But the flip side of it is, if you don't have the patent, then how can you persuade somebody to invest all the money that you need to develop the product and get their money back? And there is a lot of money needed. So what's the open source argument? The open source argument is that you don't need all that money because the community can develop whatever it is you're talking about. And I think in some circumstances that is true. What I've seen with open hardware projects in the life sciences space, and there's some really cool ones which come out of places like Cambridge, and some that I've watched, for some years. And there is one catch, which is that with an open source project, quite often people want to buy it. And then you're back into this same old thing of how do you recover your money? If you invest a lot of money to mass produce something and quality control it and sell it, it's probably going to cost a lot more than if the people built it themselves. And then the question is, will they pay that much more? If it costs £50, the robot that we developed, the parts cost less than £500. But there's no way that we could make and sell it for £500, because somebody has to put it together and check that it works and write the software and fix the software and fix it when it breaks and deal with the customers. OpenTrons is a really good example. For those of you who haven't heard of OpenTrons, they have an open source pipetting robot, but they sell products. And so they must have somehow come up with a way to protect enough of what they sell. And maybe it's just selling it. I mean, maybe that's all you have to do is sell the product. If anybody else has got any thoughts on this, I'd be really interested. But I would love to develop technology fully open source. I think it would be brilliant. I think in the clinical diagnostics area, it's almost a moral imperative that we do that. But I don't know how you get people to persuade them to make a factory and build things and sell them. I don't know. I mean, having said that, lateral flow technology is pretty much open source now in terms of IP. There's lots of patented lateral flow modifications, but it's just a strip of nitrocellulose with some colloidal gold and some antibodies. So there's other forms of IP than patents. So yeah, if anybody's got any thoughts or experience, I'd love to discuss that and share it. But my feeling is that it's all difficult. It's definitely worth doing, though. I'm not saying it's a bad thing.
Do pharmaceutical companies or governments sort of sponsor products?
So I mean, we have done this. So government funding is a really good route. We've found that to be very successful. Although we often promise how much money we're going to make in the future for the UK economy. So at some point, I wonder whether they're going to check. But yes, I think increasingly we see the benefits of government investment into that kind of thing. So I'd like to see more of that. I think there's a certain irony that the Oxford group that developed a COVID vaccine very quickly, their expertise was almost entirely built on overseas aid funding for developing things like Ebola virus vaccines in Africa. And so the government then cutting all the money that's going into that funding scheme right now to, this week is particularly beautiful irony. But so, yeah, I think government funding is a really good area, particularly for things like antimicrobial resistance. Pharmaceutical companies are an interesting area because, yes, they use analytical technology on a massive scale. So I think that's a really good route. Yeah, that's a good opportunity. You have to, I think if you're looking at a pharmaceutical company, though, you have to compete with the, if you like, the big commercial players in lab instrumentation. And I think a lot of pharma companies will probably prefer to buy a closed system from a big name, a Zeiss or a Roche or a whatever, than to go to some, hippie open lab makerspace and buy their kit there. But when I spoke to a pharmaceutical company about the development we got, they said, well, we bought this amazing robot, but as soon as we the maintenance contract expired and the person left who had been trained, we've never used it. So maybe they would have been better off with an open source system that they could co-develop. But yeah, those are all really interesting thoughts.
So we have a question on the comment section talking about IP and when countries don't obey IP laws. So if you patent your idea, then it reveals what your IP is, and if the countries aren't going to obey it, that I guess injects some risk in.
Yeah, I mean, I think that what I've seen, so I've looked at developing products in India, for example, which isn't a country that may or may not, I don't know what its status is in terms of protecting IP, but we have no IP coverage there. So anyone can copy our technology in India. But I think the game there is that if we could develop a product in India, that product would be valuable in European and US markets, but we would have developed it further and nobody could copy it in the areas where we do have IP protection. So I think a lot of people are very comfortable with protecting IP just in the most significant buying countries where the IP is protected, because you might be able to copy it in certain countries, but you wouldn't be able to then export it from those countries into the countries where you do have patent coverage. So I think that I see that as a big problem. I see it's much more of a problem is the cost of just maintaining patents and getting them granted in the first place, and the huge time cost as well. It's expensive and slow, and also it depends on your area, but in a lot of areas, the patents are kind of trickery in the sense that what you can patent is very, very specific and niche, and that doesn't necessarily matter. But so I think the big question isn't where you have IP coverage or patent coverage. I see your point about publishing what you do, but that also comes under the category of often what you patent isn't actually the most important know-how to get your product to work. Almost always it's the other way around, that the detail of getting it to work is not part of the patent, in my experience anyway. But again, I'm sure it varies from field to field. One thing we haven't discussed is the non-patent IP, which is becoming incredibly important. So things like software and the software system, and obviously any data that you collect or generate, or the analysis of any data. So you can probably have a completely open system, but still be the only people that can sell it, because what you're doing is very difficult to copy inherently. And in the life sciences, that's very common. So for example, antibodies are quite difficult to copy. Well, they're not actually, but they're supposed to be quite difficult. Maybe Barbara could comment on that. Now she's working for an antibody company. Maybe we don't want to go there.
Well, I'm just in the lab for now. Well, actually, I don't know. I know they have everything very well protected, and especially, for example, myself, I work on a team that develops antibodies for other companies. And I know, especially if those companies want to use these antibodies for therapeutics, for example, they need to come to an agreement, and needs to be very well protected. But
so there are things.
I don't have much knowledge on that.
So, but I think the important point is there are things other than patents that can protect what you know. And maybe that's more important. And maybe that's what people pay for, is your ability to turn something into a product that works.
I think that's probably it. It's just having an IP doesn't mean that you can create something that somebody's going to buy, which brings me sort of to a question. I mean, you have a great technology platform that combined can do something, and you could go in various areas, and so you have examples of doing this. But what would be the killer application that's based? What are the unique features, advantages, and benefits of this technology that would be able for you to go out into the market and go and talk to either pharma companies that are using the high throughput method, or maybe it is a point of care diagnostic, but what is it that is the greatest need that requires your technology versus things that are already out there that can help you kind of develop that? Because you might then find maybe a company that wants to, you don't have to go all the way and develop the product. You might just team up with a company that has develop something already in that market, or supplies already to pharma companies, for example, that would take that technology on and develop it. But it's driven by the market need that
Sure.
your technology specifically can address that need.
So, and this is something we've been looking for a long time. So, my preferred way to answer that question is to say that I'm giving this talk as a university academic rather than as a representative and director and founder of CFT. But it's a very good question. So actually, for me at the moment, my belief is that the real urgent commercial priority for the Micro Capillary Film technology is actually to get it out as a research product, because we can do that right now. We have a website, we have product, we have boxes of, you can buy a box which has got everything you need in it, and that has no regulatory barriers, and it has no further validation. We've done tons of validation. At the moment, we've got 3 frontrunners. In the microbiology area, I think the most exciting product opportunity is in antibiotic resistance testing in veterinary applications, because we've had a lot of discussions with farmers and vets in various different countries, and we keep coming back to exactly the same thing, which is they have nothing. Mm-hmm. So there's a massive gap and there's enormous need. I'm not saying it's easy. We know what some of the problems are, but you're right. And I think the next step is definitely a proper push on partnering and business development to work with the probably microbiology companies that already sell into those markets. But I think for a number of reasons, that's a really good fit. I mean, one simple reason is that human diagnostic would be very compelling for urinary tract infection. And in some ways that's easier technically, but it's a massive, massive path to get to a point of having a human in vitro diagnostic. And I'm not entirely convinced that people actually want to do what everybody says they want to do, which is a rapid urine test for antibiotic resistance. Because at the moment, you can just take an antibiotic. What's the problem? And in the places where antibiotic use is closely regulated, the empirical guidelines work fine. And in the places where antibiotics are freely available and anyone can buy them, they're not going to use a test because they don't need to. So the human clinical is the most important global aim, but actually the veterinary one's really interesting. For immunoassays, we are, just like everybody else, we're developing a proof of concept COVID-19 assay, but there's plenty of competition there. We actually have got to a really interesting point because we finally have we've got an injection moldable little widgety device that people can use. This is the one we use for our dengue study. And the gap there is really, really simple. We just have to go and talk to all those companies, the diagnostic companies, the pharma companies. And my excuse for not doing so is for the last year, it's been a bit difficult to talk and do business development. That's not really true. I just haven't had time. But, maybe that's one of the biggest, going to my barriers slide, I put lots of costs on here and things like that. Hidden in here is business development. This takes an enormous amount of time. And it's not fundamentally difficult. You just need to go and talk to people and more people and more people until you find the right people. But it's expensive in time, and you need the right people doing that because you need people who really understand not just their technology, but they understand what the person they're talking to wants. And what you also need, which I don't have, is the sort of deal making skills. I have no idea how to do that. I'm probably quite good at selling, but I think I'd be terrible at negotiating contracts and agreeing things. So that business development cost is really significant. And the sooner you can do it, the better. But then it's cost money. So
how are you funded at the moment?
So CFT is angel investment funded. I mean, it's essentially got a single sole angel investor. But we are looking at other options at the moment, and it's complicated. But yeah.
No, it's very complicated, but I guess it's true. It's the team that either can partly do the development and understands, and also goes out and talks to people to find that killer application for which there's a need to do this. So, but it requires, as you said, you need funding to get somebody on board, or a co-founder that is equally driven and has, skin in the game, as you say.
Well, and when we have a
funding.
lovely co-founder, the problem is that his expertise is in a different sector, because his expertise is in making this stuff.
Okay.
And he doesn't know anything about life sciences. So he's brilliant and he's very, very experienced. But he's not the sector knowledge co-founder, he's the manufacturer. And actually, here's a big irony with this technology. It's really interesting because most of these sorts of technologies, one of the big hurdles you have to overcome is how to make the thing. But we're mass producing this. We can make it in the UK, 10,000 meters of the stuff. But then you have to turn that into a product and you need the business development. So ironically, we've got a manufacturer as a business angel, which is brilliant, but it also means we don't do the business development. But I think that's true of a company and any investor, that it's very hard to get absolutely everything.
And going back, sorry, I was just going to ask a question that's in the chat at the moment. So going back to MCF and the applications of the micro capillary film, someone is asking what's the shelf life and what's the robustness against environmental factors. I guess that's very important.
So
if you want to use it in developing countries.
Yes. So all of the devices that we've developed, so we know a lot about the immunoassay devices. They're very stable. And we've actually done antibody coating of this material and taken it to Thailand, and it works fine. We've shipped it or posted it or put it in our luggage or whatever. We've done stability tests. The stability of any immunoassay device depends on the stabilization of the core reagents, which are primarily things like antibodies and occasionally enzymes and reagents. So it's exactly the same as the stability profile of a lateral flow device, except slightly different. For the microbiology, you just have to be really careful with things like antibiotic assays. If you've got antibiotics in there, one of the biggest problems I know in developing countries for antibiotic resistance testing is just keeping the antibiotic sample standard quality controlled and stable. Because if your standard is off, then you don't get meaningful results. And that's true with the oldest style of antibiotic resistant test, which is just a piece of, basically a piece of filter paper with antibiotics soaked into it. So it's no different for our technology than other things. One of the things we've always strived for is simplicity, but you can't have everything. So if you want the analytical performance, you need some complexity in the system. But at the end of the day, it's just a test tube. Sorry.
I was saying that Martin had a question before me, so I don't know if you want to.
Go on, Martin.
Well, yeah, so what sort of assistance do you get from the University of Reading? And how are you balancing your academic needs with your company?
Well, they're lovely. I mean, they pay me a salary, and no, it's brilliant. The University of Reading is very different from the University of Cambridge in lots of ways. The simplest way is it's much smaller. So, we don't have a big tech transfer office. We do have a brilliant business school, but it's not, most of it isn't heavily involved in sort of tech innovation, which doesn't mean it isn't good. It's really good. We've got lots of good connections there. We've got a nice local angel group. As a location, Reading is brilliant because we have people and we've got, connectivity with everything. We're surrounded by all the big pharma companies, all of the tech companies, Microsoft, Oracle are just up the road. So it's a slightly different environment from Cambridge. We have a slightly different approach to IP. We tend to be more open to early and complete out licensing, which would never be tolerated by the University of Cambridge. So some investors really like that. And some businesses really like working with a university that's very happy to just hand over IP if you pay for it. But that's the nature of being much smaller. I think we're not that different in size in terms of the number of students, but we have a much smaller turnover and a much smaller research base, and we don't have as much funding and investment. But we do fine. It's small. And the other thing is, historically, we've always been an applied universities. So we have excellent strength in agriculture and meteorology, for example, traditionally. And pharmacy, I think, is a very good fit with that, because pharmacies, again, is applied science area. And actually, that's the other reason. One of the other reasons I'm working on with mastitis in cows is because we have 700 dairy cows in a working milk farm that sells milk to Waitrose or Sainsbury's. So we actually have a really nice setup on site to evaluate our technology, and we've got fantastic researchers working in that area. Of course, University of Cambridge has got good vet school as well. Very nice. But they don't have a farm, I don't think.
They do. It's not very big, I don't think.
Okay. Our farm's bigger than yours. But I, in a way, I see it as a continuum. We're very connected to Oxford and London and Cambridge. I know it's a bit annoying. It's on the wrong spoke, but.
Yeah. I mean, I can think of two products coming out of the CRUK. One, and the amount of money and effort that's got into trying to get them through to market. One's, I think, it's pretty much worked, but the other's had a couple of years and the money's run out. And that's, so even with all that help, I'm not sure what the success rate is.
Well, but I think that's the other thing. One thing that I think it would be great if it was more widely understood, and it took me at least the last 10 years to work this out, is that it's still worthwhile investing that money and trying and pushing forward. We have to keep doing that. That's how innovation works. It probably is quite a wasteful process, but then research by its nature is, and often it's unexpected. So I don't think we should worry about spending money and not getting there. But
I have a question.
Yeah.
So I saw your rolls of capillaries. They reminded me of optical fibers. I wanted to ask whether you kept any time optical fibers into these capillaries for any more sensitive assays or something.
It's a really good question. We actually do the opposite trick, which is one of the cool things, and one of the cause of our IP is that I didn't even mention this, but the plastic itself is a fluoropolymer. And so it has these round capillaries in it. And the reason we use a fluoropolymer is because it has a refractive index which is identical to water, which has the unexpected benefit that when you fill the capillaries with water, they become completely transparent, which is very important for analytical sensitivity of your assays. And what that does mean, though, is it means it's exactly the opposite of an optical fiber, because you have no, I mean, obviously for the overall plastic, you could use it like an optical fiber, but for the individual capillaries, you have no optical change in terms of refractive index between the water inside the tube. So in a way, it's a bit like a test tube, except for the fact that it's invisible when it's filled with water. But yeah, we have kind of played around with that, because you can, if you're doing something like a fluorescence assay, you can kind of illuminate it from the end if you want to.
Yeah, and on that note, I would like to ask another question. Now we would like to replace in photonics all optical fibers, and would like to come to silicon photonics, where we would like to put on a solid substrate, hard substrate, which is not rollable, not flexible, to reduce the size and get advantage of that. But on a similar style, for like your device, somebody can make an injection molded hard substrate based capillaries. How is that different than these roll-ups?
So the two differences with this material than an injection molded microfluidic device, one of them is the refractive index matching that I mentioned. It's very hard to mold the fluoropolymers, so people don't tend to. But what you end up with is whenever you have a transition from water to the plastic container, you can see optical distortion, and you can obviously design it to avoid that being a problem. But the other thing is that, the mass production of this with the reagents loaded is still, at the moment, probably easier and cheaper than filling reagents into an injection molded part. But I don't personally have especially if in my university role as opposed to my commercial role, I don't have a problem with injection molded devices. I think anything microfluidic has got huge potential. And now is a really good time for some of the reasons I mentioned in terms of reading out the results of the microfluidic devices, it is difficult to tool up and mass produce something injection molded. It's quite easy to do the proof of concept, but when you get up to scale, you've got to worry about whether every single part is exactly the same size. If it isn't exactly the same size, how does that affect your assay? But yeah, I'm all in favor. There's so many different fabrication methods. You've also got to think about how you assemble your actual product, not just making the plastic component that you use in your product. There's so much technology and ideas out there. Another really good trick I've seen done a lot is you can make microfluidic devices quite easily by cutting plastic sheets and then laminating them together, which gives you a different geometry, but it's very, very quick to prototype and it's very cool. Basically, you're making stickers and then sticking them together. We played with that a bit, and there's lots of people doing that. And essentially, that's what a lateral flow technology is it's a lamination of different materials.
Yeah. At which length your tubes, length of the tube is curling up? Because it looks very flexible to my eye.
It is, yes.
Yeah. So at which length it will curl up?
I mean, what we tend to do is we'll put it in some kind of cartridge, a bit like a lateral flow device, before we would actually use it in a product. So it depends on the application. For the microbiology, we tend to use little holders. I don't seem to have any lying around, but essentially, I normally have. I can print one out. We have a holder that looks a bit like this, which just has the short strips. For the immunoassay, I'm not going to show you yet because we haven't published the final cartridge design, because there's some other clever fluidics that we haven't disclosed yet. But so the flexibility is actually quite useful because you can make different shapes and play around.
Great, thank you.
There's a question. Oh, sorry.
Yeah.
Was
okay, I can go ahead if you.
Sure. No, go for it.
Okay. Hi, all. This is Varun. I was just thinking about your earlier remark about business development and trying to think about where are those lucrative applications which could make the business case more strong. A thought that popped in was often if you look at diagnostics as a market, it's always lower cost per test. I mean, that's the driver, right, for the market usually. You don't want to spend a lot on running the test. You want to optimize it. You want to adhere to regulations. But at the end of the day, someone won't be paying a lot of money for that diagnostics unless it's a very significantly expensive disease, if for lack of a better word. Have you thought about more lucrative areas where this could be applied, the technology? Because I can see like biomanufacturing could be an interesting space where you might be running assays like the ones you run for an immunoassay, but then the size of the market and the value of that market is significantly larger. So I wonder if solving similar problems that you do today for diagnostics, but in a different context, like biologics production, could be something which makes a super strong business case. I don't know what your thoughts are.
Thank you. That's a good suggestion. Something I have seen with other diagnostic companies in the past is that some of them have ended up becoming analytical subsidiaries for pharmaceutical companies, because sometimes there's more money in doing biomarker analysis for a big clinical trial of a drug, where the budget for the drug development is massive, than there would ever be market in actually selling a diagnostic into the same market, which to me is really tragic, because it seems to me that you're missing the opportunity to solve a clinical need. If you have a biomarker measurement that's clinically important for the clinical trial, it should be measured for the patients as well. And then it gets complicated. You can worry about things like companion diagnostics, but you're right, it's not always that straightforward. One thing I would correct you on, though, is that in my opinion, clinical diagnostic is not really a low value proposition, because some of these tests cost, even the lateral flow ones sell for £5+ per test, which doesn't necessarily much, but for example, UK PCR COVID testing, I don't know how much it's costing, but I think it's more like £100 per sample. That's a lot of money for one product, you know, for one piece of plastic, if you can sell that for £5 to £100. So I think in some clinical diagnostic applications, the cost that the payer will pay per test is a surprisingly high amount. So I think there is a big margin in clinical diagnostics if you can find a big enough market. Sorry, the big problem is finding a big enough volume to justify the upfront clinical trial costs, which can be quite expensive.
No, I agree. I only meant it in a relative terms of other spaces you could look at.
Absolutely. No, this is really,
totally.
really valuable. Yeah. Yeah.
And there is also a scope, right? Like supporting the business case to get the money flowing in to run the operations could allow for additional value to be created in other areas. So that's the only thing I was thinking, but thank you.
And I just have to say on this note that, well, while doing my work with Al, so my aim when writing my thesis and everything was to have the MCF as a point of care diagnostics. First on my master's for platelet function testing, for example, we were looking at measuring the level of blood clotting. And then later on my PhD, I was also looking at measuring blood biomarkers for stroke. So yeah, I think there's a big usefulness with MCF that could potentially be used in point of care diagnostics. And it's something that's easy, can be used in a lot of diagnostic tests. And yeah, maybe one day we'll be able to use it. I don't know. Maybe you can tell us more about that, but that's a big discussion, I guess.
I mean, I could talk about point of care diagnostics for another whole hour, but that's a different discussion.
Exactly.
There's a question in the comments about cumulative data gathering across disease and territories. Is there any business cases you've explored around the use of the data of these diagnostic techniques?
So, I mean, I feel kind of embarrassed to say that we've been doing this so long, but that's one of the things that we haven't done, because when we started doing this and looked at the business cases in most detail, that idea, it was a fledgling idea, but it's now obviously a very big idea. I guess where we came from the beginning was something very related to that, but different, which was this idea of a diagnostic fingerprint, because for probably at least 10, maybe 20 years, one of the biggest frontiers in diagnostics full stop has been the concept that if we can measure multiple parameters and combine them with an algorithm of some kind, we can make, and that's been through lots of different iterations. Sometimes it's been called personalized medicine, sometimes it's been called stratified medicine, more recently it's been called precision medicine. But the idea is a very powerful one, which is that if you can get enough good quality data, you can tell the difference between conditions and treatments, and also response to treatments that previously have been opaque, simply by measuring more than one parameter. And because we can measure 10 parameters, we do fit that very well. But what you're saying is that there's now a business case for upfront investment to gather a big enough database to actually have those measurements. And one of the strengths of our system that I think would fit that is that we can do point of care and high throughput lab measurement using exactly the same measurement tool, which is actually pretty unusual, because if you use an immunoanalyzer, it's a big machine which does various things, versus a lateral flow or a point of care immunoanalyzer. They actually work in different ways and they'll give you a different analytical measurement. So yeah, I would love to do that. And maybe that would be something to pitch to, we haven't really pitched for significant investment for quite a few years. Under that business development heading is actually raising the next phase of investment. And yeah, we don't have, as Martin said, we don't have a massive tech transfer team pushing that for us. But I forgot to say, actually, the original invention was a Cambridge invention when I was a postdoc there. So we spent a little bit of Cambridge Enterprise investment, and we had a huge amount of support from them, just in case anyone's seen the video. No, they were brilliant.
Anyone has any last questions? I know we are running out of time, but if everyone wants to say any last words.
One of the interesting question you posed during the talk was how much to be open sourced and how much to be patented. Did you ever consider opening up any of your technology for the open source?
I mean, in due course, the patents will expire and it'll be open source anyway. This is what I'm thinking about a lot with other projects which are new. At the moment, I don't see that it will be easy to raise investment for a fully open business. But as discussed, this idea of data collection, things like that, that if you can develop hardware and get it widely adopted and develop know-how and data, then you don't need traditional patents, because you have, and actually in some areas, things like this is already happening. Interesting, this is already happening in microbiology. So there's one of the major changes in analytical biology in a clinical lab is that a lot of clinical labs use mass spectroscopy, or is it spectrometry? I can never remember which one it is, MS we'll call it, to fingerprint bacteria. But the sort of what I think is the stealth clever bit of that is that yes, the big companies get to sell their MS instruments, but what people don't necessarily realize is that they're feeding the databases endlessly with data. And the databases are absolutely essential because getting a fingerprint of bacterial protein fragments never tells you what the bacterial species is unless you have access to the database. So in a way, that's a double whammy, that they've got the instrument costs, which are huge, and the maintenance costs, which are huge, and they've got the data as well. But I think that's completely closed. You were asking a question about open source. Have I thought about it? Yes. Have I done anything? Not yet.
Okay. Okay. The complementary question is, you saying something along in your presentation about using consumer products like hardware, for example, to develop some of the new concepts or devices. Did you ever think about what could be the barriers of using them?
Yeah, it's a really good question. So, everybody's been saying this for at least 10 years. Let's use our smartphones for diagnostics. But what happens if you're never going to get regulatory approval for something when you don't own the system? Probably, I think you can, but it's a long story. Normally, a clinical diagnostic product is a closed system which is evaluated closed, and it's got a product definition. So imagine if you're using an Android or Apple smartphone to take a picture and then somehow analyzing it in an app and then giving a diagnostic. What happens if it's a different version of the operating system and the camera is slightly different? I don't see how you can sort of close that circle. But that doesn't stop the hardware itself from being really cheap, because not that long ago, buying digital cameras was really expensive, and it just isn't anymore. So you can buy and especially through things like the Raspberry Pi Foundation's cameras and other examples like that. So I think the best option is to use the hardware from a consumer product, but not to use a consumer product for a clinical diagnostic.
Yeah. Okay. Just to comment on my own question on your answer, one can use this colored matrices as a stickers, and we can take with
yes.
and that can do the color correction. That I don't see as a problem with. We can somehow balance the.
Yes, exactly.
Of the camera, but I haven't seen cameras used for any real-time application. Coming back to the mastitis thing, you mentioned there are iPhone adapters released hardware products. I don't know if they are being used by farmers anymore. Did you ever see them, somatic cell counters?
Yeah, no, that's a cool idea. I haven't looked for that recently. Yeah, you're right. For things like somatic cell count, coming back to your point about taking a picture with a control, I think that's very important. So in a way, what I see that is that's a bit like getting a human user to look at a color and choose it, but you're doing that electronically, which I think is brilliant. So you're getting around, you're not trying to write the software on the computer, sorry, on the phone to control the phone camera. You take a picture of your sample with something next to it, and then you're comparing, is it the same color or a different color? So that's a really good way around the hardware problem. I mean, yeah, so there's lots going on there. It's exciting and things are developing very quickly. Although I do think it's wonderful that, you know, in the UK, we're doing this mass testing. Everybody's getting all these tests to use at home, and they're all read by eye. You just look at it and say, yes, it's positive, or no, it's negative. And bear in mind, whoops, the big driver in point of care diagnostics in the last 10 years has been lateral flow readers. So all the companies that make lateral flow will sell a reader, and the readers are mostly either a smartphone, Android or similar, or a tablet that's been OEM'd into a reader, which has got its camera and some software. So there are so many lateral flow readers out there, which would make it easier to systematically analyze the results of lateral flow rapid tests for something like COVID-19. And we're just using our eyes, which I think is brilliant, which goes to show all of that investment was maybe misplaced. I don't know.
Yeah, there was a very good argument on that. The thing is that most pregnancy tests, only 75% true, accurate, because people, when they're under anxiety, they misread the tests. But when they keep it in these readers and they can clearly say pregnant, no pregnant, yes or no, the accuracy level jumped up to 95%. That's not because anything to do with the underlying technology, but people's anxiety. Maybe they're selling
and even just the lighting and things. The great thing about a digital camera is it will take a picture and analyze it, and it will be the same every time. Whereas if you get a human to score it, somebody's going to get it upside down, somebody's going to get it the wrong way around. It's not, as people say, it's really easy to use. It's not that easy to use, and there's a huge amount of evidence for that. But lateral flow is the gold standard at the moment in terms of mass producibility. And as I say, we mustn't underestimate the power of having huge factories that can mass produce these things.
So a couple of so firstly a couple of plugs. There's so Ward's put up a link for the Make Space every Friday. There's a talk about startups and early stage companies and talking about challenges. So if you're interested in these kind of topics, definitely take a look there. There's an email to talk to and a link to go to. Also, the University of Cambridge is always looking for things and inventors and projects to do for research by doing. Also a link there from Katia. There's a question at the end talking about other topics for biology clubs. So if you were, say, running a talk every once a month on entrepreneurship, what kind of questions would you be asking?
Yeah, we always gather just after these events. We stay around usually for a few minutes after the presentation just to discuss the next event. So if you'd like to stick around or join the biology club, you can just stay on this Zoom video call for a few minutes. And if I don't think anyone has anything else to say, here is your chance. Okay, so yeah, thanks everyone for joining. Thanks all for this great presentation. It's always a pleasure to hear what you have to say, even after 5 years. Something like that.
We must catch up sometime.
Yeah. For sure.
Maybe I can get discounted antibodies.
Rating is not that. Sorry.
Maybe I can get discounted antibodies.
Yeah, well, maybe. And yeah, so if our next event is in 2 weeks, so you can check on Meetup what's next. We usually do this every 2nd and 4th day of the month, as I said before. And yeah, thank you for joining. Have a lovely week. And thanks all once again. Bye.